Yeah, thanks, Dare. And like I said, I was sitting watching. Oh, yeah. No, I can holler with the best of them. But like Dare said, I got to sitting and watching he and Matt's presentations yesterday and realized that great minds think alike, and we'd really converged on the same set of thoughts about genomics as they relate to EPDs and EPD accuracy. So the tweaks that I've made here, I'll reinforce a number of things that Dare and Matt both dug into with just a little bit more. I think that repetition is really important for learning these things. I don't learn something until I've heard it about 30 times. 3 times will have to be good enough on a couple of these topics today. But I really want to talk a little bit as well about the other functions that genomics play in the seed stock realm. So we'll talk a little bit about genetic defect testing. I'll very briefly mention parentage testing and its importance. And then we'll dive into both sort of the commercial genomics. I know this is a seed stock symposium, but commercial genomics, I think, is something that I'm getting more and more questions about. And some of the lessons that we can learn from the commercial application of genomics and how that differs from what we're using genomics for in a seed stock setting, I think, is really important. And as your commercial customers bring you questions about, hey, should I go out and spend money to genotype my heifers? Or I have genotyped my heifers, how do I utilize this information to make mating decisions? Sort of how we can walk through those things together and learn again a little bit about seed stock genomic testing as it relates to commercial genomic tests. So with that, I'm going to dive right into the 3 main functions. And again, these won't match your proceedings. I'm sorry. They're more just different ways of putting Matt and Dare's presentations together. So this is all new and fresh. But 3 main uses of genomic tests. The first is parentage verification. Before genomics was genomics, we were utilizing genetic markers to verify parentage, right? We were utilizing those to make our pedigrees, the pedigrees that we use to calculate EPDs, as accurate as possible. The pervasiveness of parentage errors throughout these pedigrees was and still is a major challenge for us in delivering as accurate an EPD as possible, right? We talked about the importance of phenotypic recording, consistent phenotypic and accurate phenotypic recording. You can have the most accurate, in-depth set of phenotypes attached to an animal, but if it's incorrectly attached to other animals in the pedigree, it's not of particular use, right? So this is a big part that I think gets overlooked. If we can correct these pedigree errors before they become a problem through parentage verification, the genomic test has served a really big part of its overall utility. The second is genetic defect testing. We'll spend a little bit of time talking about this because I think it's an area of genomics that, again, we've known for a long time that we can put these simple genetic defects on a genotyping test and avoid carrier matings. But as we start to think about some of the other harder to see, harder to measure genetic defects that come in the form of what we call lethal haplotypes, genomic tests on populations become really, really important for identifying, predicting those, and avoiding carrier matings. So something that I think we can learn a lot from the dairy industry on. They've done an exceptional job of managing these lethal mutations and these genetic defects in their population. So again, a big part of what genomic tests do for us. Outside of the thing that we talk about the most in the context of seed stock populations, which is how do we enhance the quality of our genetic predictions, make our EPDs more accurate through genomics. So we'll hit on all 3 of these. The first, again, parentage verification, very simplified. What we're doing, and this is a small example, right? We've got 3 potential sires. We know the genotype of the calf. We know the genotype of the dam. We utilize these parentage markers to go in and do a process of elimination, right? If we've only got 1 parentage marker, we probably can't eliminate any of these 3 sires, right? Because that calf is a big A, little a, dam is a big A, big A. If the sires all have the potential to pass on that 1 little a allele, we can eliminate them based on this marker. So the reality is that as the number of markers that we use for parentage scale up, we can become more and more confident through this process of elimination that, hey, This marker at locus D is the one that allows us to distinguish the parentage between these two calves, right? This is pretty straightforward. In reality, as we look across thousands of animals, we have to use much more than 5 of these loci in order to identify parentage. But again, it becomes relatively trivial for us to add these on to a genomic test, and that serves a big part of how the breed associations are able to make sure that their pedigrees are as accurate as possible. So again, that's a very important component. I'm only going to spend one slide on it, and then we'll move into genetic defect testing. So genetic defects are something that we've known about for a long time. The way that they arise, particularly in seed stock populations, mutations happen, right? Regardless of what animal there is. When they happen in the germline, right? So they happen where those sperm cells, where those egg cells are being generated, right? That's how they get passed down to offspring, right? So I've got mutations that are happening in my skin cells, in my blood cells, in my eyes, every organ, right? Mutations are happening because DNA replication isn't perfect. But they only get passed down to offspring when they happen in the germline tissues, right? So particularly as we think about bulls and how some AI sires in seed stock populations have a really, really big footprint in the breed, okay? We might have thousands or tens of thousands of offspring that come from a very, very popular high genetic merit bull. The way that genetic defects in some of these, what we call deleterious mutations, right? Mutations that occur and they break something, all right? Most mutations don't do anything, but the ones that break a gene, right? That gene might, we can think about dwarfism, right? We break some sort of a gene in a growth pathway, and all of a sudden those animals don't perform in the same way, right? Some sort of a developmental gene being broken by mutation, that can cause lethality of that calf that's born. Or the more challenging thing for us to hunt down are when those happen, a gene breaks that's very important early on in embryonic development. And so when that happens, it just shows up like a decrease in fertility. All right, that's a mutation. And again, these things tend to be recessive, so we can think back to a Punnett square. If you only carry one copy of, say, the developmental duplication, right? So this is going to put an extra leg maybe out of the middle of a calf's back. If you only carry one copy of that, you're not going to be affected, okay? It's only when we match together 2 carrier animals, and 25% of those matings between carrier animals will come out as little d, and that phenotype expresses itself. The important thing to keep in mind, these tend to be pretty rare in the populations, okay? But as we think about seed stock populations and we think about, line breeding when it works and inbreeding when it doesn't, right? So the more we try and stack together favorable genetics, if they're related to each other through shared descent, right? We call this identity by descent. If they've got the same mutation here, because they trace back multiple times in the pedigree, we cross them to another animal who has multiple pedigree connections, right? As we increase those number of genetic connections, as we look back into that animal's genealogy, the likelihood that we're going to match up these unfavorable mutations that occur increases substantially, right? That's why we don't typically see this in commercial cattle, but we certainly see these genetic defects pop up more commonly in seed stock populations. And the important thing is that they get driven to a significant frequency, a frequency to where we start to actually see them pop up in the population because they occur in high genetic merit animals, right? If mutations only happened in bad animals, we wouldn't see any of these things pop up. It's the fact that that genetic defect, right, is tied to an animal who is genetically superior. He gets used a whole bunch. That drives the frequency of that deleterious mutation up substantially. So as these new genetic defects become apparent in breeds, oftentimes we can go and do some detective work back into the pedigree and find that animal where the mutation occurred, and more often than not, they had very, very large footprints on the breed, right? So again, this is sort of some background on genetic defects. The important thing to keep in mind from a genomics perspective is that I think there's a tendency for our initial thought when we hear that we have a carrier animal is to throw the baby out with the bathwater, right? We're not going to use them, okay? We're not going to use a carrier animal. I don't want any of that, right? The reality is that through genomics, right, when we're genomically testing and we know where these mutations are, we can breed around these things, right? We're not going to get affected animals when we mate, if we don't make carrier animals together, right? So I think the dairy industry, again, if you look at a dairy sire book, they've got dozens of these things up at the top of every page, right? They know what their defects are. They know how to map them very quickly and effectively when they pop up. They include them on their genomic tests, and they breed around them, right? There's no way to find a non-carrier animal for every single genetic mutation that exists in the dairy world, right? So what they do is they take that into account when they're designing matings. And again, I think we can do something very similar in these beef cattle populations as we need to. So the challenge is finding some of these things that aren't very apparent, right? So if you've got, again, a developmental duplication, right? That calf's got an extra leg coming out of his back. If you've got curly calf, which is it's lethal when that calf is born, okay? It's straightforward enough for us to say there's something wrong with that critter and we need to turn in this information to our breed association. So that's my one piece of advice about genetic defects to a seed stock population. If you see something weird, right, the worst thing that you can do is take that calf out to the dead pile and say, I'm going to wash my hands of this, okay? Breed associations have a huge vested interest in tracking these things down, designing genetic tests for them, and giving you a tool back to avoid making these matings that would result in a defective calf, right? So again, the best thing you can do when you see a genetic defect pop up is to report that to the breed association. And what they're able to do is they work with a variety of academic collaborators, and we're able to make this genotype to phenotype association, right? So as you get a defective phenotype, we can oftentimes, with enough genotypes, work backwards and find that gene, find that mutation that's driving that simple recessive disorder in the calf. The challenging thing, and I think sort of the next frontier and where genomics is really going to come in to help, is, again, if there's something that's lethal at this stage of, say, a blastocyst level embryo, right? So this is just a few days into development, a lethal mutation occurs, right? And we match up two unfavorable homozygotes, and that thing just dies, right? It dies in the cow. We're never going to see a phenotype that we can report back to a breed association. It's just going to show up as a decrease in fertility, right? But that still is very much something that we'd like to know about so that we can avoid putting matings together that are destined for 25% of those carrier matings to show up as lost fertility and that calf not go on to develop. The way that we track those down through genomic data is we try and find places in the genome, right? Find genetic markers where there's a depletion of homozygotes, right? So we might have homozygous big A and lots of heterozygotes. So that says to me that, hey, this animal is competent when they're homozygote. Having one copy of the mutation isn't bad enough for that embryo to cease development. But when you match two of those little a's together, you never see that in the population. That tells me something about that being a genetic defect in the form of what we call a lethal haplotype or an embryonic lethal mutation, right? So this is sort of the next frontier. I think that a lot of breed associations have taken up that search in earnest to find these. It turns out it takes a lot of animals in order to find these, especially when they're very, very rare. If you only see a lethal mutation in 1 out of every 10,000 animals, right, it's very, very hard to track down. These things, due to their decrease in animal fertility, typically don't rise to super high frequency. So again, this requires lots of genotypes on lots of animals and the ability to go in and find those that don't have any of those homozygotes around. So that's the main thing that I'll say about these genetic defects and parentage testing. Any questions along these lines before I change gears? Yeah, I know that, and I don't know how much this is common knowledge. Angus is working on this, I think, to some degree. Matt, anybody else that's taken up this search in the same way as Angus?
No, not at a breed association level.
Yeah. Yeah, so again, Angus would be the main breed that is looking for these actively in their data sets. So, and again, the way that they're doing that is exactly like this, trying to find those genetic markers, those stretches of DNA that just don't show up in the homozygous alternative form in their population. But again, it's really hard to track those down, and it's hard to, again, just probably too deep in the weeds, but with our sparse genotype arrays, getting those down to a single mutation is quite hard. You got a big block of DNA that you see never shows up as a recessive homozygote, but trying to dial that down to a single marker to test is tough for a number of reasons. But that's a major initiative that they're in the process of working on at the moment. Yeah, so the genomic test, when you buy a 50K or 100K genomic test, it's going to have those known markers for all of these Angus specific, and the same thing could go for a Red Angus, a Hereford, or whatever. If there's a known mutation in that breed, there's a greater than 99% chance that it's on that genotyping array, and that's where they're doing that genomic test for the presence of one of those mutations. And those lower density assays should still have your genetic defects on there, right? So it's I guess that's the trade-off, right, is having that information on the front end with the first pass of genotyping versus having to go back and generate, again, if you have an AI sire or something, a maybe 100K test that includes these markers on that subsequent array. I didn't realize that those ULD, ultra low density chips aren't including any of these, so that's that's a challenge for sure. Right, and that's the that's the thing to notice here, right? And that's a great point, Dare, that when you look at the primary breed or breeds of incidence, it's very rare that these mutations have popped up in multiple breeds in the exact same spot, right? That's sort of like lightning striking, 10 times in a row on the same day in the same place, right? It's not likely that you're going to see those pop up. So crossbreeding from a commercial perspective, that's why we don't deal with these. If we're making F1 crosses, the likelihood of us getting, a curly calf is very, very low as long as we're not breeding, an Angus and a Red Angus, right? Where you can maybe still have some of those mutations that float around, right? So again, crossbreeding is the solution from a commercial standpoint to avoid these genetic defects popping up in the commercial herd. All right, anything else here? All right, I'm going to keep moving and talk just a little bit about the justification for why we need the genomic tests that we do. We break our traits out into these two main classes. The first are our simple traits. These are things you can draw a Punnett square for. In knowing those two parental genotypes, we can predict the proportion of those offspring phenotypes as well as genotypes, again, because one gene sort of rules them all. That's the strategy that we use for these genetic defect tests, right? We know that this is simply inherited. One gene causes this phenotype. So again, designing a test that can predict the phenotype of an animal is really straightforward. The reality is that almost all of the traits that we care about are complex, right? They're polygenic, many genes, lots of genes spread throughout the genome. Some are large effect, but oftentimes even this is what we call a genome-wide association. So we look all across the genome and we ask, is this place associated with an increase in this trait value? All right, this is for stature, which is one of the most complex traits. It's very highly heritable, but there are hundreds to thousands of genes that are involved in stature, whether you're a human or a dog or a cow, right? Some of them, so PLAG1 right there, it's a gene that we know is of quite large effect, but even the biggest effect genes for a complex trait have explain a small amount of that overall genetic potential that an animal has to have a large phenotype in that case, right? So again, small amounts of variation, generally across a lot of different genes. So what that means is that from a genomics perspective, we have to be able to capture genome-wide variation, right? It's not good enough to just look at these handful of genes. You can think about the gene star markers, right? We found these handful of markers, or the T-SNP test for fescue adaptation, right? These are just a couple of markers that were found from what we call a candidate gene approach, right? The reality is that the traits that that they were saying they were predictive of are super, super complex, right? even the best of those markers, which most of them weren't even good at all, explained a very, very small amount of the overall phenotypic variation. So in order for us to capture that, we have to go out and genotype as much of the genome as possible, right? We have to have a sufficient number of markers to capture all of the genetic variation that's captured in that genome that can relate to these complex traits. So again, that's my, I guess, justification for genomics, and then we'll dive into some of the nuts and bolts of this, right? Again, nothing that is new compared to what Dara and Matt said, but genomics are an essential tool for us, right? We've got to use them alongside all of our other strategies for sire selection, but what they do is they help us estimate that genetic merit more accurately, right? A genomically tested animal, the EPD that comes out, right, sometimes it goes up and sometimes it goes down, right? But we'd expect that the average of what happens after we get a genomic test to be 0, right? You're going to get some favorable, you're going to get some that decrease in overall value, but the important thing is that that value is always more accurate than a non-genomically tested animal, right? Genomically tested animals are, the EPD that we get out is always better than what we had before, even if that trait value has gone down. And so what that allows us to do is increase, again, the accuracy, particularly of unproven animals. Right? These are animals that we don't have the luxury of seeing multiple calf crops worth of data flow back into the genetic evaluation. We're more confident in those, and particularly as your commercial customers, they're more confident that the animal that they're purchasing based on EPDs, that those EPDs actually represent the true genetics that they get to pass down to their offspring. And the important thing, too, to keep in mind about genomics and genomically enhanced EPDs is that they accelerate population genetic gain, right? Occasionally we get these animals, and I get far more calls on animals whose EPDs have gone, again, in the opposite direction after genomic tests. Nobody calls me when things work out, I guess. But on average, right, the accuracy of selection that we increase when we utilize genomics moves that full population forward. And that's the thing, as we were having the discussion, which I thought was really productive after Matt's talk, is that, again, these tools are designed for population improvement, not individual animal improvement. And I think we sometimes get caught up seeing the shifts in individual animals rather than what that overall accuracy does to the improvement of a population. If we were pig breeders, right, and there are a couple of companies in the world that breed pigs, right, their measurement of what genomics does to their EPD accuracy is very different, right? Because they're looking at the population scale rather than a handful of animals that are a part of this larger population. So again, genomics, in increasing the accuracy of which we do selection, moves this population forward, right? What it doesn't do is predict phenotypes for individual animals, right? It only captures our statistical estimate, that animal's genetic potential that they can pass down to their offspring, right? So again, we talked about all the other moving pieces that go into that in phenotype that gets expressed. Genomic tests, right, again, they only give us an estimate of that animal's genetics they can pass down to their offspring, not necessarily of individual animal phenotypes, and they're not able to capture any of the environment or what we call non-additive genetics that factor into an animal's overall genetic potential in a single generation, right? Just what can get passed on from parent to offspring. So with that, I think that the way I always like to frame genomics is they help us, again, reduce the risk of our commercial customers' bull purchase, right? I put this up in every presentation that I give, I feel like. The bull purchase is one of the riskiest decisions that a commercial buyer makes, right? They're making an investment in the future of their herd, right? Not just in the immediate term, but if they're keeping replacement females, it's multiple generations, right? So again, we need to take as much of the risk out of this as possible, give them the tool that is the most predictive of how that animal is going to perform from passing, again, that genetics down to the offspring in their herd. And so genomics do a couple things, right? They help us reduce the number of train wrecks that occur. So again, that your commercial customer is out looking for a calving ease bull, making sure that the bull he buys is truly a calving ease bull, right? That's the big one, and that's the train wreck that I'm sure some of us have gotten calls about, right? When that expectation doesn't match the actual genetics of that animal. right? And so again, the more accurate our selection tools can be, the more confident we can be when we're marketing that bull as a calving ease bull, right? As a bull that's going to add some maternal good, right? We want to be confident that those EPDs are doing that so that our commercial customers don't get into a train wreck downstream of that bull purchase, right? The other things that they do both within our herds, so as seed stock producers, we're in a race against other seed stock producers to try and make the best animals, right? That's really what it's about, is how do we make genetic progress towards an overall breeding goal faster than our competitors or faster than the rest of the population. Genomics allows us to do that, again, just as Matt described, because they help us make our selection decisions more accurate. So again, the closer we are to selecting based on that animal's true genetic merit, which is value we can never know, right? But we can use these statistical estimates in the form of EPDs to get a lot closer to what that animal's true genetics are, right? And so the more related those two things are, the more often we're able to make the correct genetic selection decision. The more often we make the right decision, the faster our genetic changes towards our breeding goal, right? And assuming that our breeding goal is in a positive and profitable direction, we're helping our commercial producers be more profitable faster when we make that genetic selection decision more accurately. And so, again, the things that we have to do in order to get there is tease out that environmental variation, right? We can't pass creep feed down to offspring, right? So utilize our contemporary groups to pull out that place-to-place environmental variation, management variation, get everybody on a level playing field. And then we utilize EPDs to estimate what this genotypic proportion is, right? What's the heritable piece that a bull can pass down to his offspring in the form of genetic potential. And so the key thing and where genomics comes in, and previously where having progeny flow into the genetic evaluation came in, is contemporary groups allow us to pull that environmental variation out, right? But that's only half of the problem. The other half is we have to figure out which genetics an animal inherited from its parents, right? I loved Dare's example of his dad and his son and those chromosome paintings that he did. That's exactly what we're trying to do, either through progeny testing or through genomics, is figuring out which chunks of DNA an animal inherited from its parents, and hopefully those genetics that they inherit from the parents are favorable ones rather than unfavorable ones. So again, with low amounts of information, so if we don't have any progeny information, we're flying basically from these animals' own performance, so that helps us understand a little bit, right? And their parent average, right? So if we looked at parent average EPDs without any own performance, that's, it's better maybe than doing phenotypic selection, but not by a whole lot, right? That own performance can tell us a fair bit about an animal's genetic potential, because if he way overperforms our estimate from parent average, that lets us know that maybe he got a favorable shake when it came to the random sampling of genes, right? But the reality is that the true genetic merit of an animal, that possible change again that Matt talked about, that can be pretty large when we have limited amounts of information on animals, okay? The true genetic merit might be between those two horizontal points on each of the white and orange EPDs, which means that we could see a reshuffling, a re-ranking of these animals from a true genetic merit standpoint rather than their EPDs. The solution previously is that we add progeny to the evaluation. We get a more confident estimate of which genetics those animals inherited from their parents, right? So that's ultimately what's happening, and I'll give an example here in subsequent slides. Again, a very similar figure to what Dare talked about in his own family, but here's 3 full siblings, and anybody who's done embryo transfer, right, they've made ET full siblings, knows that when you do a genomic test, the EPDs of these animals can change pretty considerably, right? And it's all about that assumption of 25% relatedness to the grandparents. That's not genomic reality. Genomic reality, you're still getting 50% from dad, right? There's still 50% of each of these bulls who has that red and yellow combination. They got 50% from dad, but which 50% they got can differ quite a bit. And when we talk about these traits, these complex traits, having genome-wide effects, right? There's a good chance that you can inherit a whole bunch of really good genetics, or you can inherit a bunch of bad or inferior genetics, and that's going to differ based on the proportions of those parental genomes, right? So if the maternal grandsire of this full sibling group is an absolute rock star, this bull is probably going to be the genetically superior of those, right? All of our genetic progress is built on this idea of the random sampling of genes and identifying the animals that got the more favorable shake when it comes to that random sample. This is a figure I like from my PhD advisor, Jared Decker at the University of Missouri, that shows an example of the billions of possibilities just based on the 30 pairs of chromosomes that cattle have, right? All the different possibilities you can get just due to the random way that those chromosomes line up during the process of meiosis, where sperm cells get made, right? You, in theory, could get 100% of that sperm cells chromosomes could come from this bull's sire, right? The blue chromosomes, all the way to 100% from the dam, right? So again, the randomness ends up being billions of different combinations. We need to be able to track these, again, knowing that there's the proportion of the quantitative trait genetics that are on these chromosomes is going to differ a fair bit. The way that this sort of works in practice, right, is that an animal's underlying genetic potential is a function of both of his parents and dumb luck, right? This dumb luck term is what we call Mendelian sampling. Again, that was mentioned earlier, but it's the random sample of genes, right? And for complex traits, that Mendelian sampling component can take up about 50% of the overall genetic variation can just come from that random sample of genes that you get from an animal, right? And this is going to differ from population to population, but that number can be quite large, right? And that's what explains these differences in the genetic merit between siblings. And so our goal in animal breeding is how do we, as accurately as possible, estimate what that Mendelian sampling term is. And again, going back to our example here, we see an increase in the accuracy of an EPD as we add more progeny because we're getting more confident in what this Mendelian sampling term is. Again here, the idea that you've got this parental expectation, right? This is our parent average in EPDs. If we've got a bunch of calves out of a bull and they're consistently overperforming this parent average, what that in reality is helping us do is estimate this Mendelian sampling, right? This is saying that this bull probably got a good random sample of genes from his parents, right? And he's going to be more likely to be genetically superior and pass down that genetically favorable inheritance to his offspring, right? If there was no Mendelian sampling, right, and we were just mating together good animals, right, and all the calves were exactly the same, if they got 50% of their parents and none was better than the other, we'd never make genetic progress, right? All genetic progress is built on this randomness, and so our job as breeders is to identify the animals that got the more favorable shake from this random sampling. And so what genomics allows us to do is identify this earlier rather than later, right? There's that trade-off, just as Matt said, in breeding to a proven sire, right? You're giving up some time in terms of genetic progress, right? If we only bred to bulls that were 99% accurate, it takes even an AI sire 3, 4, 5, 6 years to get there. What's happened in the meantime is that the rest of the breed has progressed, right? And all of a sudden there are way better animals that exist out there from a genetic perspective than that bull who is 100% accurate, right? We're very confident in his EPD, but there's been a lot of genetic progress that took place in the time that it took him to get there. And so there's this trade-off. So how do we find the young animals, the unproven animals that are genetically superior without them having multiple calf crops, right? And so genomics, in allowing us to directly observe the DNA that an animal inherits, gives us a better prediction in the form of this EPD, right? We reduce that possible change that you could see in an EPD without that bull ever siring a calf, all right? And I'll emphasize again later on, phenotypes are still super critical for us to build the accuracy of these EPDs. But from a commercial perspective, right, this gives me much more confidence in that bull as I'm going out and making a purchase, making an investment, that he's not going to cause a train wreck and he's going to help me advance my herd because I'm more confident in this EPD before he's ever gone out and sired a calf. you guys are familiar with how genomics happens, right? So I can skip through this. But the foundation for how we design these genotyping tests, I think, is important to go over. And it's built on this idea of linkage disequilibrium is our scientific word for this. But basically what this is saying is that we don't need to go out and genotype all 3 billion places in the cow genome, right? Because DNA gets inherited as chunks, we're okay to go in and genotype one place in that chunk and use that as a representation of the neighboring pieces of DNA, right? So a mutation, or we'd say that little red dot is causal. That's the mutation in the genome that causes a change in our phenotype. What that means is that we don't have to go through and genotype each one of those markers because there's a statistical association between that mutation that causes a phenotype and these other places in the genome that we maybe have a marker. Over time, that's going to break down, right? There's this phenomenon of recombination, and again, we won't get too far into the weeds here, but that breaks down over time, which is, I think, justification for continually collecting phenotypes so that we can maintain the relationships between our markers, right? So maybe we have a marker at point B up there. We maintain that statistical connection between where our marker is and where the mutation is that causes that change in phenotype. And so again, this is really nice because it allows us to take a reduced set of markers when we design a genomic test and utilize that to account for these big chunks of DNA that get inherited together because of their physical proximity to one another. And so again, I won't go too far in depth here, but this foundation, right, so the machinery that we're still using to calculate genomic predictions actually came out about 10 years before widespread genotyping did in our seed stock population. So in 2001, this paper comes out that says, hey, if we could effectively genotype all of we call them effective segments, big chunks or haplotypes of the genome, with about 50,000 genetic markers, we could do genomic prediction. The tool doesn't come out until about 2008, and not till in 2010, 2011 are genomic evaluations getting rolled out. But again, this allowed us to go in and find the 50,000 best genetic markers, so they're evenly spaced, we get that full genome coverage, and they account for the most genetic diversity possible. So we call these moderate to high frequency, right? We want to see as much variation in these markers as possible. What that allows us to do is go in and account for all of the effective segments that exist that are related to these complex traits and develop genetic prediction models that integrate those in. And at the end of the day, another progeny equivalent table here, increase the accuracy in unproven animals to the same degree that a full calf crop's worth of information would flowing into that genetic evaluation. So really quickly, I'll walk through a couple of the genomic prediction strategies, right? We won't go too far in depth here, but want to give a survey, compare and contrast just a little bit from breed to breed how EPDs are getting calculated and how they're largely equivalent. But before that, I can't emphasize this enough to seed stock audiences. If we're not collecting accurate phenotypes, these genomic prediction models are worthless at the end of the day, right? If we're not putting together informative contemporary groups, right, these genomic predictions are not effective. Genomics doesn't solve all of our problems. It helps make our selection tools better, but it absolutely requires that we do a good job of collecting phenotypes and making sure those flow back into our breed associations. So 2 flavors of genomic prediction. The first would be this model developed at the University of Georgia called single-step genomic best linear unbiased prediction. It's a mouthful, right? Single-step GBLUP. And the idea here is that single-step GBLUP functions about exactly like a pedigree-based genetic evaluation, but what the genomics does is it helps us identify the relationships between animals better, right? So instead of assuming that 25% relationship between an offspring and its grandparents, we can actually go in and utilize DNA sharing, right? How many chunks of DNA effectively do these animals share with one another? And that allows us to correct some of those pedigree relationships to better reflect, well, I guess, biological reality, right? And I'll give some examples there in a second. And again, the single step part of that is blending together this genomic piece with information from that wider pedigree, some clever statistics that they figure out on how to do that I won't go into a ton of detail on, I guess. The second is a hybrid marker effects model. So this, anybody who's familiar with Bolt, so IGS breeds, an American Hereford would run a Bolt model, which uses marker effects. So it estimates the effect on a phenotype at this particular marker across a genome-wide set of SNPs, and then comes back in and accounts for additional variation that isn't captured there based on the pedigree, and is able to do that in a single-step manner using some, again, clever math that I won't dive into today. But the important thing to keep in mind about these before I go into detail is that from a statistical perspective, the assumptions that get made in both of these models, they're equivalent, right? There's no inherent difference, right? When we go back to the theoretical derivation of all these models between single-step GBLUP and a hybrid marker effects model. Execution is different, but if we make the same assumptions at the start, they're effectively equivalent models and will almost always lead us to the same point at the end of the day. So I'll start off with single-step GBLUP. The only change here, you'll notice, we're still having progeny records. We're still having animals' own performance going into the calculation of the EPD. The only difference is that instead of using just pedigree relationships to figure out this covariance between the animals in the population, we're utilizing a combination of genomics and pedigree there. The reality, the only thing that changes in how we calculate a genomic EPD versus a pedigree EPD is how we, again, compensate for that kinship term in the mixed model equations where EPDs come out of. The difference here, right, this is a little toy example of one focal animal, its parents and its grandparents. Again, we have these expected pedigree relationships. On average, if we have tens of thousands of animals, that'd be about the average relationship between an animal and each of its grandparents. Genomic reality is that this animal in particular is more related to its paternal grandam and maternal grandsire than it is to its maternal grandam and paternal grandsire, right? This is how, again, that random shuffle of genes, we're able to pick up these differences and realize genomic relationships between animals. We're able to identify animals maybe that aren't related in a pedigree sense, but they share DNA with one another. Accounting for all of these in our EPD calculations ultimately makes those more accurate, right? So that's one way that we can use genomics. The other, and this is the hybrid marker effects model, we can estimate effects at each one of these places in the genome. So having a B is maybe worth, a B allele here is worth having an extra half a pound of weaning weight. We can do that all the way down the genomes of these animals. And then ultimately what we come up with is a marker effect estimate of that animal's genetic potential. Again, some clever math to correct for pedigree differences and other variation that isn't contained in these markers. But at the end of the day, they get us to a similar point as that single-step GBLUP model would have gotten us to. So again, I'll leave us with this thought before I dive into one last little topic, and that's that regardless of whether EPDs improve or decline as we add genomics, it's always better than an ungenomically enhanced EPD, right? We're getting ourselves closer to the truth. We're getting more confident that that value is representative of an animal's true genetic merit. And genetics work when we look at the population level, all right? So this is something that Kelly Ritalik from Angus Genetics Incorporated reported on here at the World Congress of Animal Breeding this past summer, showing the differences in relative genetic gain before and after genomics across all of these traits. And to me, that's the ultimate example, right? Is that when we make a more accurate selection tool, our seed stock producers make better genetic selection decisions, they make better matings, and that genetic progress increases at a different rate, okay? So you'll notice the directions of some of these is different. We've actually decreased a little bit of milk. Again, milk is best in moderation. Marbling is held steady as Angus was already exceptional in marbling. But for these other traits where genetic selection is heading in an upward direction, ribeye area and weaning weight and carcass weight, you see a significant difference in the rates of genetic change in each of those three traits after the arrival of genomics. Yeah, I'll emphasize that again and again and again. You can't build good genetic prediction models without phenotypes, no matter how you slice it. We're really good at genotyping animals, so Angus is getting nearly 200,000 new genomic tests a year, but so the average increase in the accuracy of an EPD is obviously good on the individual animal level, but Angus would much rather have a bunch of carcass phenotypes come in than one more additional genotype, right? And so I always talk about phenotyping in the context of this phenotyping paradox, right? Where the difficulty of measuring a trait is sort of inversely related to the number of phenotypes that we have in an evaluation. Birth weight is the phenotype that a genetic evaluation has the absolute most records on. I can almost guarantee that. Because the barriers to measuring that, right, are having a hand scale and a little bit of bravery to go out and pick up a calf that's just been born, right? This is an easy phenotype to collect. We have a lot of records on it, and that means that the selection tools are quite accurate. The same goes for weaning weight, a little bit more of an investment. We have to buy a platform scale to measure that. But as we get to other traits that either go down the supply chain, so carcass traits, I know any breed association would love to have true, real carcass traits on offspring of animals from that breed, right? Those get harder to measure, which means we have less of them, and the quality of the genetic prediction it sort of suffers from a lack of phenotypic information. Then as we move into feed efficiency, this is again, one of the big things. We know it's important, but measuring those, the barriers to measuring a feed efficiency trait are pretty substantial, right? You have to go out and invest in a feed intake system, which is probably not practical for almost every seed stock operation. And then we talk about the future of genomics, the future of genetic prediction and all that. I think that probably the things that we're more interested in lay even further out here on the difficulty or price of measuring these traits, right? If we don't have a selection tool for a trait that's economically relevant, it's probably because it's hard to measure. And so I think that a lot of our future work in the genetic space is actually going to be in the phenotyping space, okay? How do we find ways to measure things like bovine respiratory disease incidence, right? That's a big problem that we'd love to crack, but that's a really hard trait to measure, especially subclinically, right? Greenhouse gas emissions, direct measures of metabolism, environmental stress tolerance. If I could get a comprehensive measure of fescue adaptation, I'd be a millionaire in this part of the world, right? But it's hard to measure, it's multifaceted, and so this is where a lot of our work, I think, is headed, is how do we model and get these things into a genetic evaluation. So I'm up on the end of time. I've got some slides to talk about commercial genomic testing, but we'll leave that for another day. The most important thing is that we'd really love to have you guys down in Knoxville next year for the Beef Improvement Federation's annual symposium. We're excited to host this. Matt mentioned that at the end of his presentation, but June 10th through the 13th in Knoxville. BIF, I think, is one of the organizations that I think is the most useful and informative and practical. I'd say it's a third academics, a third breed associations and industry, and a third producers, right? I learn something every year that I come back from, very boots on the ground oriented, and how are we advancing genetic selection within the beef cattle industry. It's where I got to know Matt and Darren and a bunch of the great producers across the country. Having it in our backyard is a real treat. So I hope that you guys will come down to Knoxville. Always informative, days packed with science and producer perspectives, and a cool tour of some East Tennessee operations that we think are really innovative in this genetic prediction and genetic progress space. So again, we'd love to have you there. We'll have some more information coming out over the subsequent months as we get a little bit closer to the day of, but we're excited to have this in the backyard and hope that you'll make the trip over to Knoxville to join us. So with that, there's a couple of takeaways and some contact information from me, if I can be of assistance to anybody else. And I think we are ready for lunchtime. Yeah. All right. Sweet. That's bonus content, huh? All right. So again, this is the thing I've been requested to talk about more by county agents, by producers in Tennessee. This is a hot topic, and so I want to give a little bit of extra context to commercial genomics. Again, whether this is you testing commercial heifers, your customers testing commercial heifers, just a little bit of the, what this does, what it doesn't do, and how it's similar and different to what we're doing in a seed stock context when we talk about genomics. So I put this slide up here again, and I think it's especially true when we think about commercial heifer genomic testing, which is typically our use case for this in a commercial setting. The most important thing that I want to point out here is what genomics don't do versus what we need a commercial heifer genomic test to do. It doesn't predict phenotypes for individuals, right? It just predicts an animal's genetics that they can pass down to their offspring, all right? So if we think about the most important thing to get a handle on in a commercial heifer setting, right, we need longevity. My economist friend Charlie Martinez would tell me that cow longevity in a commercial herd is the most important trait to my bottom line, okay? And so what genomics, again, don't do is they don't tell us what exactly that heifer's phenotype is going to be from a longevity perspective. This is a lowly heritable trait. 10 to 15-ish% of that trait variation comes from genomics, right? So we can capture that with a commercial genomic test maybe, but there's another 85% of that comes from development, from environment, and other non-additive forms. So again, if we think about a crossbred commercial replacement, there's a lot of non-additive genetics that play into that animal's phenotype that gets expressed, which is ultimately what saves us money at the end of the day. All right? So that's just a little bit of extra context about what these commercial genomic tests do versus what they don't do, right? They're not going to tell us exactly how this animal's going to perform. They are going to tell us something about that animal's genetic potential, but that comes with a couple of caveats again that I'll add here. I think there are multiple tiers of commercial genomic tests. So the gold standard would be to have your commercial animals in a genetic evaluation, right? And so, for example, the total herd enrollment option at American Simmental, this allows a commercial registration option. So all of the UT cow herds are enrolled in Simmental's THE. They get a full set of genomically enhanced EPDs back. They're treated just like an animal, just like a purebred Simmental animal would be in that genetic evaluation. Red Navigator does something similar with Red Angus influenced animals. So those are sort of for commercial producers. They're keeping records like a seed stock producer would, right? The barriers to entry there are higher than most seed stock or most commercial producers rather are able to invest, right? It takes me a lot of man hours to make sure our total herd enrollment is up to date on 5 separate herds totaling 1,200 or 1,300 cows, right? It's a heavy lift. The other more, I guess, approachable and commercially available genomic tests, I'd divide into straight bred. So these are for high percentage Angus animals, the GeneMax Advantage, and Brangus has a similar commercial product. Again, these are just for animals that are of high genetic proportion of genetic ancestry from a single breed. And the second class would be these multi-breed commercial tests, definitely the most common, and these will be useful in breed composition of about anything that's not Bos indicus influenced, okay? So again, these are probably our most common: Igenity from Neogen and Inherit Select from Zoetis. I won't go into the nitty-gritty of all this, but a couple different ways that those two commercial genomic tests are calculating the values that they report. But the most important thing to keep in mind about a commercial genomic test and really as we think about genomics in general is that we do this process of training and then the testing population that we use the training data for, all right? So in order for these commercial genomic tests to work, we have to start from a reference population. So these are animals that have a genotype and they have a phenotype. And what we go and do is we train each of these markers. We figure out what we think the effect at each one of these markers is based on animals that we have phenotypes and genotypes from. And then in the commercial sense, we're utilizing those SNP effects to test animals that don't have phenotypes recorded, all right? And so then we utilize that, what we call a molecular breeding value. Again, this was straight from Dare's presentation. This value that we've gotten from this training population, we're predicting into this set of unphenotyped animals. The important thing to keep in mind here is that this is very, very reliant. Our ability to accurately predict a marker effect is very reliant on how related the testing population, so these commercial heifers, how related they are to the population in which these marker effects got trained. If you're trying to use a reference population of Angus animals to predict into a Charolais Hereford cross, right, your prediction accuracies, those SNP effects, are going to be very, very bad, right? The more related this testing population is to the training population, the higher quality those commercial genomic tests are going to be. This is absolutely an essential component of all this. I say all that, but these things do work, right? They are predictive. Again, if we look across a big population of animals, this is some work from the University of Missouri that takes a DAMS GeneMax score. So this is the commercial Angus test, and it asks, so based on the DAMS GeneMax score, what does the offspring phenotype out of each of these animals look like, right? So what you'll see is that there's a significant slope to each of those orange lines, right? The DAMS GeneMax score is significantly predictive on average of those calves' performance. The issue is that there's still a lot of spread here, right? This it's serving its purpose, but again, it's not perfectly predicting phenotype of those offspring. Some of these dams, right, they're way underperforming that line of best fit. Some are way overperforming. Again, you've got this big amount of spread, and that's because these things aren't perfect predictors of offspring phenotypes, right? Just of those animals, the genetic component of each of those traits. If we went to something like longevity, right, some of these even lowly heritable traits, there's an even bigger spread around the predictability based on those GeneMax scores versus the actual calf's phenotypes. The one other thing I've done some simulations on here is really just showing the effect that a bull purchase has on a commercial operation, okay? And so what we've got here is effectively 5 generations of selection in a commercial herd. So our green line is a commercial herd that randomly selects their replacement females. The red line is a commercial herd that genomically tests their commercial females. And the blue line is the seed stock population at which those two different commercial herds are pulling their genetics in from. And so as we measure genetic progress over time, which would be the slope of that line, right? The line is more vertical. We're making more genetic progress over a fixed amount of time. What you'll see is that the 3 slopes of these lines are identical, okay? That tells me that these two, right, regardless of how we're selecting replacement females, most of that genetic progress is coming from the seed stock bulls that we bring into our herd, right? And so my general recommendation to commercial herds interested in genomic testing is, for one, I think you can get a little bump here, right? This sort of bumps you up a little bit from a genetic progress perspective from where you start. But the long-term genetic gain that creates versus what's created by the bulls that come in and have a bigger footprint on your herd, I think that bull purchase far outweighs what a commercial genomic test can do on the replacement female side, because we're much more intensively selecting those bulls that come into a herd versus the replacement heifers. We just can't put nearly as much genetic selection pressure on a replacement heifer than the seed stock industry can put on the bulls that they're marketing. So my suggestion, right, is if you're a commercial herd and you want to test 20 heifers, right, at $40 a pop, that's $800. So the issue comes, are you spending an extra $800 on this bull, or are you buying as good a bull as you possibly could? Because I think that from the long-term perspective, that's going to generate more genetic progress over the long term than again, getting those selection decisions more correct on the heifers. There are a lot of other moving pieces to this, but again, I think that commercial genomic tests might work in some settings. But as far as a blanket recommendation, I'm a little bit more hesitant there. Again, so much of this genetic progress is going to come from the bull side. Anything you'd add to that, Dare?
No, we talked about it a little bit yesterday. I mean, another big deciding factor on the return on investment is going to be how that commercial producer markets his calves.
Right.
If he's just selling weaned calves, which most of our guys do, it's going to be a lot harder to pay for the test than if he's taking them all the way through and selling them on the regular. Probably the biggest comment.
Yeah, no, and I think the other place where you can see a return on investment of these commercial genomic tests, you get that advantage again in your selection, the selection decisions you make at the heifer level. If you can turn around and use that average value of the heifer crop to market your steer crop, that's where I think there might be a little bit of an advantage there. You might see a return on investment there bigger than what you'd see on increased accuracy of selection on the heifers alone from a genetic gain perspective, right? So if you can use that extra information to market those calves in an added value way, again, based on their genetic potential of that whole calf crop, then I think that the commercial genomic tests can make a little bit more sense and more clearly pencil out. But anything else, Matt, any other thoughts on that too while we're sort of on dinner? In a lot of cases too, it's a function of, well, I tested the heifers that I was already going to keep, right? That's just confirming a decision that's already been made. You've got to use it to inform decisions, right? It's just like an EPD, right? You've got to use this as a selection tool, as a decision support tool. Otherwise, it's just extra information floating around. So testing, if you're going to utilize commercial genomic testing, you've got to genotype more heifers than you plan to keep. If you're just, again, genotyping the heifers you already know you're going to keep, you're confirming what you already know, and that doesn't move the needle at all from a selection accuracy perspective. Yeah, no, I think that's part of the sell on these things, but it again comes back to from an ROI standpoint. That's a phenotype, right? Is ultimately, and so these things aren't predicting phenotypes; they're predicting genetic potential. So for highly heritable traits, again, if you've got some of these end product traits that are very, very highly heritable, these predictions are going to capture a little bit more of that variation. Whereas for cow longevity, where the accuracy or the heritability of that trait in a purebred population is maybe 10%, right? Then there's another 90% that go into that. We're only capturing a small piece of that with the genomic test. But 90%, right, is coming from how that heifer is developed and managed. Yep, no, I lump fertility in as probably the biggest component to longevity, right? That's your number one culling reason in a commercial herd is that she's open, or it should be. So yeah, fertility is huge, and you can break that out into a number of different, subsequently more difficult rebreedings, right? Heifer pregnancy is one thing. On fertility, yeah, fertility is another one of those complex traits, right? It's lots of variation spread across the genome, lots of small effect mutations that affect it, and another one where it's lowly heritable in addition to that, right? So that's where I think the conversation about EPDs, right? It shouldn't be like it's slowly heritable, we can't change it, we should just do environmental manipulations, right? I think that lowly heritable traits are where it's most useful to have a selection tool, because you can still move that genetic potential for a lowly heritable trait, but it's a lot harder to do with a phenotype. And for female fertility or cow longevity, the bull that you select never has that, right? So it allows you, again, an EPD gives you a tool to select for a trait that you'll never see observed in the animal itself. So that's where you get the biggest advantage, I think, of an EPD is on a lowly heritable trait that you can't measure in the animal that you're selecting ultimately.